Okay, hello everyone, welcome to the class. In today's class, we'll be beginning with the topic vision and we'll be covering various aspects related to vision. We'll be looking at it as a sensation, how transduction happens, and what is the structure of eye and various brain regions associated with it. So, let us begin with it. So initially, first of all, we look at how various sensation transducts, how they reach from being receptive to the sensory receptors till the brain region that is there. Right. So we know that we have various types of sensory modalities. Right. We have vision, we have audition, olfactory, gustatory, and tactile. Vision is associated with the sensory organ eye, audition with ear, it is for hearing, olfactory is for smell, nose, gustatory is for taste, that is our tongue, and tactile is for touch, skin. Now, first of all, reception will happen, which means that our sensory receptors or our sense organs will receive the sensation, will receive the stimuli. Now it can be of any modality, right? Today we are going to focus majorly on vision. Now there are specialized neuron in our sensory organs, that is eye or other organs. We have certain sensory receptors which will receive those information and transduct it. Now what is transduction? Transduction is the process by which the conversion of the stimuli is there. Now whenever we are typing, we know that computer does not understand a b c d or any word that we type. They have their own language. For example, if we talk about binary code, it is 0 and 1. Same, our brain does not understand the visual, the auditory, the olfactory information as it is. So we need to convert this information into a language, into a way that our brain will understand. And what does our brain understand generally? Our brain will understand the electrical impulses, action potential is there, or it will understand the chemical impulses, neurotransmitter, hormones, etc. Right? So in transduction, we will be converting this sensation, this sensory information into a electrical impulse. So till now our sensory organ received the information, and with the help of these specialized sensory receptor, transduction occurred. Now these sensory receptors will relay the information to our central nervous system and specifically to the area of thalamus. Thalamus is known as our relay center. All the type of sensory modalities except for olfactory, they move from this, or they move in this particular direction itself. Thalamus is the relay area which would, further on it is like a manager, right? So all the information is coming to a manager. Now the manager will relay the information to the person who needs to work on that information, to like which part that is relevant. Same, thalamus will be relaying the information to the exact brain area that is necessary, that is relevant, or that is associated with that particular sensory modality, right? So except for olfactory, all these sensory modality will move through thalamus. Also, one more thing that we need to discuss here, that all the sensation except for olfactory are contralateral, which means that you all must have like heard about something like that. The things that we do from our right hand get processed into the left brain and so on and so forth. That is the contralateral function, right? The functions are switched, and the the smell olfactory sensory modality is ipsilateral in nature, which means that there is a direct, the information from the right nostril will be transmitted in the right brain and so on and so forth. You can understand it like that. So all the sensory modality except for olfactory will move from thalamus and they will be called contralateral in nature. Now thalamus will relay the information to the main projection area. Okay, now what is projection area? Let me add another slide. Now, this projection area is the primary, or it is the central area which will be important to that sensory perception, important for that sensory reception. For example, if we talk about vision, primary visual cortex is there. Okay? Now this particular area is specifically devoted to one particular sense, that is their projection area. And now the projection area will further on transfer the information to the association area. Association area basically takes information from various modalities, various sensory modalities, and integrate them. Okay? So as we begin with vision, we are going to look at all of these processes, how the reception will occur in the eye, where transduction will be happening, then moving from thalamus, projection area, and then at last association area. Right? Now moving to the main topic, vision. We know few basics. We know that sensory organ related to it is eye. Now the energy that we get, as I told you that our brain only understands the electrical impulses, but the energy that we get from the sensory organs, or that we get from this visual information, that is electromagnetic. Okay? And we convert this into electrical impulses or our action potential. Now we are going to look at other basics, such as you all must have, if you have gone from a eyesight testing, you all must have seen this particular chart at your opta like clinics and all of those things, right? So generally we define our vision in terms of 20 by 20 or 6 by 6. Now what is it? This particular chart was made by Snellen, and this particular chart was developed to assess the visual acuity, which is the ability to see the fine detail, to see the minute fine details, right? So this basically means that 20 basically resembles the 20 feet that a typical person can read this smallest line from 20 feet. Okay? And 6 by 6 is the equivalent of it only, which is in meter. Okay? So it is the perfect or the clearest vision that a person can have. So 20 by 20 or 6 by 6. Now the vision or the everything that we see or the colors that we see is perceived in terms of wavelength, and those wavelengths are denoted or measured in terms of nanometers. Now we have various kind of high altitude wavelengths, low altitude wavelengths, but our eye has a particular threshold. Okay? And we cannot receive information, or we cannot perceive information before that threshold or above that threshold. So our mind, or like you can say our eyes, are capable of getting information from 380 nanometer to 760 nanometer. Okay? So before that, there are ultraviolet rays, X-rays, gamma rays, which are very short waves. They don't have a higher wavelength, and our eyes cannot perceive it, they cannot detect it, and we have higher wavelength only. So we do not just get or we do not perceive each and everything. This is the small area or the small pattern which ranges from 380 nanometer to 760 nanometer. That is the only thing that we perceive. Okay. Now you all must have learned this in your childhood, the Vibgyor or the rainbow colors. It is the same. Now violet is the smallest wavelength wala color. ठीक है, and red is the maximum wavelength wala color. This is the whole chart, the whole scale, and there are other properties of this particular wavelength also which help us in the perception of our light waves, and we are going to discuss their physical attributes as well as their psychological attributes. So there are major three physical attributes: length of the wavelength, amplitude of the wavelength, and purity of the wavelength. Now looking at the length of the wavelength. Length is the difference between one wavelength to the another wavelength. Okay, as we just discussed, the length of the wavelength will help us in determining the color. If the wavelength is around 380 nanometer, we will perceive a violetish bluish color. If it is around 700 and more than 700, we will perceive a reddish kind of a color. So length actually helps us in determining the hue or the color. Okay, what color the object is of or the stimulus that we are perceiving is of. Then amplitude. Amplitude basically refers to the height of the peak, how high the peaks are. Okay, so if the peak is more high, it will say that it will like we will understand it as the object is much brighter. Okay, so low amplitude will means that there is low brightness or less brightness, and high amplitude will mean high brightness. Then purity. Purity is what? Purity is the number of wavelength. Okay, so if there are more number of wavelengths, it would tell us that the color is really rich, the color is really saturated, or with the richness of the color. Okay, these are the few basic properties of the light waves: hue, brightness or saturation, and it is corresponding to the length, amplitude and purity. Okay, now we'll be moving forward by understanding about the structure of the human eye and how this whole process happens. So I'll be making a eye here. Alright, so basically this colored area of the eye is the iris. Okay, and this black part, this hollow part is our pupil. Now our eye is surrounded, it is covered with cornea, which is an aqueous transparent substance which surrounds the eye, right? Then this is iris is the colored part of the eye, and this is pupil. Pupil is the area through which the light wave will enter our eye. Okay, and then the light wave will reach to the lens that is inside the eye. Now this lens is joined with these ciliary muscles which will determine the curvature of the lens. You must have seen that our eyes actually accommodate itself. Like for example, you have to look at something really tiny, you will squint, right? You will squint your eyes. That is what that your ciliary muscle or these oculomotor muscles are actually moving, and they are changing the curvature of the lens in order to adjust the in order to adjust the light entering, or the in order to see the visual or the vision properly. Now, after this, the lens will project the particular light wave onto the retina. Okay? Now, on the retina, we have our photoreceptors, where the transduction will actually happen. Okay? So, here we have two types of photoreceptor, cone cells and rod cells. So here we have a yellow spot that is known as fovea, and we have a blind spot. So fovea is also known as the yellow spot. Okay, here in this particular spot, all the cone cells are there. There is no rod cells at all. And in the blind spot is that you will not be able to see anything. So here in the blind spot, neither cone nor rod cells would be there. Now we'll elaborate more onto cone cells and rod cells and what are they. Let me add another slide. So cone cells and rod cells. These are two kinds of photoreceptor which will help in the detection of or which will help in the transduction. Okay, here the transduction happens. So rod cell will be necessary for the dark vision. This is the photoreceptor which will help us in perceiving, or which will help us in seeing the objects in the dark. Okay, cone cells they would work in the light vision, which means that rod cells are more sensitive to light and cone cells are not as sensitive to light. Okay, so in the cone cells there would be a chromatic vision, colors, everything we'll be able to see. But in rod cell there would be a black and white kind of a vision. Like for example, later at night you do not see everything clearly, right? You have a very blurry kind of a vision. Okay, like not blurry but not as clear as it would be in a full lit room. So this cone cells are made up of a substance named as photopsin, and rod cells are made up of rhodopsin. Okay, and there are various types of cone cells further on. We have S cone cell, M cone cell, and L cone cell. Okay, what does this mean and why have I written it with a particular color? S basically means the cone cells which helps us in sensing the light waves with the shortest wavelength. Now initially we learnt about this whole chart, 380 to 760, and that violet is the shortest wavelength and red is the longest wavelength. So similarly in the cone cells we would have three types of cells: S cone, M cone and L cone. S cone would help us in the perception or help us in sensing all the wavelength which are bluish in color, violet, blue, indigo, etc. Medium cone cell, M cone cell, will help us in receiving the cells which are, or the lights or the colors which are related to green color. Okay? And then L cone cell will help us in perceiving the colors such as red, yellow, orange, etc. Okay? So this is about the photoreceptor. So what have we learned till now? We have received the information, transduction will happen on this particular sensory receptor, and now what will happen? The information will move to the central nervous system, right? Now how will that information move? Let me change the slide. So you can learn the movement initially. PH back. Okay, this is the mnemonic that you can use. So P stands for the photoreceptor. So we have two kinds of photoreceptors as we learned. Then the information will move on to the horizontal cells, H, then bipolar cells, emicrine cells, ganglion cells. Okay, so PH back. You can learn this is the moment in which the movement of the electrical impulse will move and it would reach the optic nerves. Okay, now these optic nerves will take the information further to the CNS or to the brain. So this is our optic nerve. Okay, now we have two eyes, and if you would have seen that, like if you would look at a particular object closing your right eye and then closing your left eye, you would see that there is a little difference between what you see. So both of our eyes have different visual fields. Okay, so this is our right eye and then left eye. This is the visual field of our right eye and then this is the visual field of our left eye. Okay, so both of these eye would have, left eye would also have a left visual field and a right visual field. Same goes for right eye also. It would also have a right visual field and then a left visual, a left visual field and a right visual field. And this will form later on our retina. Okay, so the information, as we talked initially, that it is contralateral in nature. So the information received from this particular visual field, from the left visual field of the left eye, would form here in the right optic nerve kind of area, and then this here. So you can see that there is a contralateral processing which is happening, and we would have two kind of nerves here. Wait, let me plug in the charger. All right, so now we know about it. So both of these nerves, optic nerves, will move on or will transmit information further to our central nervous system, our brain. Now here, these nerves will actually have a layover which is known as optic chiasm. So the right side of the optic nerve will, and the left side of the right eye. So right side of the left eye and left side of the right eye the nerve will have a layover. That is known as optic chiasm. It is nothing, it is like a wire is laying and then above that another wire is passing on. That's what it is. So optic nerve, and then there is a optic chiasm. Then the information will move to the lateral geniculate nucleus, that is in the thalamus, which is responsible for the processing of the visual information. Okay? And now thalamus will do what? Thalamus will transmit that particular information to our primary visual cortex. But before that, the information also passes on from two areas, that is superior colliculus and optic radiation. Superior colliculus is a part of our midbrain or brain. So here this will be necessary for our visual reflexes. Okay? And then it would move on finally to the primary visual cortex, which is the projection area for vision. And in the individuals whose primary visual cortex will be damaged, they will have a cortical blindness, which means that in the eye, like they have no problem in the eye is perfectly fine, lens, everything is perfectly fine, retina is also so everything till here is fine, but because of the damage in the primary visual cortex, the person will not be able to see. That is known as cortical blindness. Okay, but still, as we talked about superior colliculus, it is responsible for what? It is responsible for our visual reflexes. Reflexes is what? Reflexes are unconscious. So still, even though they have a cortical blindness, still their superior colliculus will help in producing some kind of unconscious visual functions, which is known as the blind sight. That even if the person is blind, still they have some sensation. Okay, still they are able to produce some kind of visual function, which is known as blind sight. All right, moving ahead to the, so till now we have learnt about all of this area. We have learnt about that reception has happened in the eye, transduction has happened at photoreceptors, then the information moved to thalamus, specifically in the lateral geniculate nuclei, and then it transmitted the information to the projection area, that is primary visual cortex. Now the information will move to association area, and we have two kind of visual streams for that. Let me change the slide. Right, so we have two kind of visual pathways which leads to the association areas, the areas which will integrate information from various senses. Okay, so we have a dorsal pathway and we have a ventral pathway. Okay, I'll add on another slide and explain what is dorsal and ventral, and then we would move ahead further by discussing about it. So dorsal. So for example, this is the cow. So now this, like if you look at it, the above area, this above area would have a little kind of a different skin. So the above area is known as dorsal and the downward area is known as ventral. If you are going to look at your hands also, you would see that the skin on the upper hand and on the palm is a little different. Okay, so the upward is known as dorsal and the downward is known as ventral. Okay, so with this we'll now understand about this. So we know ventral means downwards, dorsal means upward, above. So the information reached to the primary visual cortex. Okay, and from here the information will move on to, it can move on in two particular areas. Dorsal area is also known as the Weir pathway and ventral area is also known as the Watt pathway. Okay. Now I have looked at a particular object. This information has reached to my primary visual cortex. Now I need to know where that information is. Okay, what is the location of that particular thing that I am looking at? Okay, what is what do we say, what is the motion of that particular thing? Okay, is it coming towards me? Is it moving away from me? Is it moving in what kind of direction? Okay, and in ventral pathway we will be understanding what object is it, what is its color, what is its shape, etc, etc. So here in the ventral pathway, identification of faces, objects, color, shape will be happening, and in the dorsal visual pathway we will be understanding the where, the location, the motion, etc. So as we know, there are four lobes in the brain: frontal, parietal, occipital and temporal. So the area in the lower region of the brain ventral region are a part of the ventral visual pathway. So it will move the information from the primary visual path, from the occipital lobe to the temporal lobe. ठीक है, and specifically we have a particular area named as infernotemporal cortex, which is necessary for the identification of objects, images, faces, etc. ठीक है. Now moving to the dorsal pathway. What will be there in the dorsal pathway? Dorsal is upward. There are two lobes which are located in the upward region of the brain, parietal lobe and frontal lobe. So here the information will move from the occipital lobe to parietal lobe and frontal lobe, and it is necessary for where? Location, motion, and specifically there is an area named as posterior parietal cortex, which is associated with this spatial awareness. ठीक है, so these are few association area. Now one more aspect of dorsal pathway. We know what is frontal lobe associated for. Frontal lobe is necessary for problem solving, planning, etc., etc. Now this dorsal visual pathway will also be guiding us. For example, we see that an object is approaching us. ठीक है, there is an object which is approaching your eye. So now what will you be doing? You will be able to plan that you have to maybe move backwards, you have to close your eye or do whatever. So this is also known as how pathway. That where you are able to know that where the location is, you are able to orient yourself, the motion, etc. But you are also able to guide yourself, and you are also able to plan in advance ki what to do. ठीक है, so I will be adding another slide. We have two visual pathway. We have dorsal pathway and ventral pathway. Dorsal is associated with the upward or above, and ventral with the downward. Dorsal moves from occipital lobe to parietal and frontal lobe, and ventral में occipital to temporal lobe. Dorsal is also known as where pathway, and now also as how pathway, and ventral is known as what pathway. We also learnt about two major regions which are there in dorsal and ventral pathway, which was in dorsal posterior parietal cortex, and here infernotemporal cortex. ठीक है, so this would be, dorsal would be necessary for location, orientation, ठीक है, motion, planning, and ventral for identification. So that was it for today's class. There is one more thing that I would like to add on. In the primary visual cortex, we have feature detection cells. Okay, so we have three kind of feature detector cells: simple, complex, and hypercomplex. Simple cells are necessary to know the orientation, कि we are lying up front, we are standing, we are sitting, कौन सी location है, orientation. Complex will be necessary for orientation and movement as well, and hypercomplex will be for orientation, movement, and shape as well. Okay, so this can also be asked. So that is it about eye. We have learnt about various aspects related to it. We learnt about how the reception of the information occurred. Photoreceptors were there in the eye, rod cells, cone cells, and there were three more types of cone cells, S cell, M and L cone cells. Then transductions happened at those particular cone cells. Rod is for our dark vision, dark adaptation, and cone for light adaptation. Then the information moved to our central nervous system. There was initially pH back, and then from optic nerve to optic chiasm, then lateral geniculate nucleus, which is a part of thalamus, then to the projection area, primary visual cortex, and then association area. We learned about two pathways leading to our association area, dorsal pathway and ventral pathway. And that was it for today. Thank you so much.